Let us continue with the process of transcription. Transcription, in the case of prokaryotes and eukaryotes, it's different. So let us first learn about transcription which is taking place in prokaryotes. In prokaryotes, the synthesis of all the types of RNA, that is mRNA, tRNA and rRNA, it is catalyzed by a single RNA polymerase. So a single DNA dependent RNA polymerase synthesizes or it catalyzes the synthesis of all the three types of RNAs. And the steps involved are initiation, elongation and termination. In initiation, the RNA polymerase enzyme, it binds to the promoter site of the DNA. We know promoter is the site or it is a sequence of DNA where the transcription is initiated. So the RNA polymerase enzyme which polymerizes the ribonucleotides to form an RNA chain binds to the promoter site. And when it binds at the promoter site, it causes an unwinding of the DNA double helix. We can see an unwinding taking place. This is the unwinding. Okay. So the unwinding takes place. This is the initiation process. So where the RNA polymerase binds to the DNA, there to the DNA at the promoter site, the unwinding of the DNA strands takes place. There is an initiation factor called as a sigma factor which is a part of the RNA polymerase. And this sigma factor, when it comes and attaches to the RNA polymerase, then the transcription or the RNA synthesis initiates. So this is the initiation process where the RNA polymerase comes and binds to the promoter which is situated at the 5' end upstream of the structural gene. At the site of binding, unwinding of the DNA strands takes place. And when the sigma factor, which is part of the, which is the initiation factor, comes and attaches to the RNA polymerase, initiation of the transcription takes place. Next is elongation. The RNA chain is synthesized in the 5' to 3' direction. As we told earlier, in the earlier module, the RNA polymerase, DNA-dependent RNA polymerase, catalyzes the polymerization in the 5' to 3' direction. So we can see the formation of the RNA chain. The free end is the 5' and the end which is attached to the RNA polymerase is the 3' end. So RNA chain is synthesized in the 5' to 3' direction. And during this process, what is the substrate? It is a ribonucleotide triphosphate. And it is having dual role. One, it is acting as a substrate. And the second, it is providing energy for the polymerization to take place. And the nucleotides are added in a complementary fashion. That means if in the DNA strand, it is adenine, then what will be attached? What nucleotide will be attached to form the new RNA chain? It will be uracil. If it is cytosine in the DNA, then in the RNA, the RNA chain complementary, the base pair is the guanosine. So in the complementary fashion, the base sequence is added and thus the RNA chain is synthesized. This is the elongation process. And termination. The initiation factor was sigma factor, whereas for termination to take place, there is a factor called as the rho factor. When the rho factor comes and binds to the RNA polymerase, and before that the sigma factor releases from the RNA polymerase, and when the rho factor comes and attaches, it brings about the termination of transcription. So this is the processes which is taking place in the transcription of prokaryotes. A single RNA polymerase catalyzes the synthesis of all the three types of RNA, mRNA, tRNA and rRNA, and the three steps are initiation, elongation and termination. We know bacteria are prokaryotes. That means they do not have the genetic material enclosed within a nuclear membrane. They have the nucleoid which is present in the cytoplasm. In the case of bacteria, the transcription and translation. We know transcription is the synthesis of mRNA and translation is the synthesis of proteins based on the information which is present in mRNA. So transcription and translation is coupled. That means translation begins before mRNA is fully transcribed. We can see this is the mRNA which is formed. This is the mRNA which is being formed. And at the 5' end, which is being formed initially, translation can take place. Both can be coupled together. mRNA requires no processing. As we told, it is polycistronic. It is having a continuous sequence of coding sequenced genes. So transcription and translation may take place in the same compartment because there is no separation between the cytoplasm and nucleus. So clear, this is the case of transcription and translation being coupled in the case of bacterial cell.